Welcome to this mini lecture from Team Biotech Wizipur. NMR is a widely used technique in chemistry, physics, and materials science to study the properties of molecules and materials. It's based on the magnetic properties of atomic nuclei, which act like tiny magnets. NMR allows us to gain deep insights into the structure and behavior of molecules. The technique is powerful because it can provide detailed information about the chemical environment of atoms within a sample. This information is crucial for understanding the properties and behavior of a wide range of materials and chemical systems. Nuclear magnetic resonance, or NMR, is a powerful analytical technique used to study the structure and dynamics of molecules. It relies on the magnetic properties of atomic nuclei, specifically their spin. NMR involves exposing a sample to a strong magnetic field, causing the nuclei to align. Radio frequency pulses are then applied to perturb this alignment, leading to the detection of a signal that can provide valuable information about the sample. NMR is a versatile tool used in various fields, including chemistry, biology, and material science, to gain insights into the molecular structure and behavior of substances. In the early 1940s, the nuclear magnetic resonance (NMR) phenomenon was first observed, laying the foundation for this powerful technique. In 1946, Felix Bloch and Edward Purcell conducted the first successful NMR experiment, demonstrating its potential. Throughout the 1950s, NMR spectroscopy emerged as a valuable tool for studying chemical structure and dynamics, finding widespread applications. The 1960s and 1970s saw the development of Fourier transform (FT) NMR, which revolutionized the field and made NMR spectroscopy even more powerful and versatile. Over time, NMR rapidly progressed from a theoretical concept to a widely applied technique, finding significant use in fields like chemistry, physics, biology, and medicine. The key principles of nuclear magnetic resonance (NMR) are the nuclear spin and the resonance condition. Many atomic nuclei have an intrinsic angular momentum called nuclear spin, which generates a magnetic dipole moment. This magnetic moment interacts with an external magnetic field. When the energy difference between the spin states matches the energy of the applied radio frequency radiation, the nuclei absorb energy and transition between spin states. This is known as the resonance condition. Understanding these fundamental principles is crucial for understanding how NMR works and how it can be used for various applications, such as chemical analysis and medical imaging. The atomic nucleus possesses a fundamental property called spin, similar to a spinning top. This spin generates a magnetic moment, effectively making the nucleus a tiny magnet. The spin and magnetic moment values are unique for each type of atomic nucleus. This nuclear spin and magnetic moment are the foundation of nuclear magnetic resonance (NMR) spectroscopy. NMR exploits the interaction of these nuclear magnets with external magnetic fields. The spin quantum number, I, determines the number of possible spin states for a nucleus. For example, a nucleus with I equals 1/2 can exist in two spin states, spin up and spin down. These spin states have different orientations of the nuclear magnetic moment relative to the external magnetic field. The Larmor frequency is a fundamental concept in NMR spectroscopy, representing the frequency at which a nuclear spin precesses in a magnetic field. This frequency is directly proportional to the strength of the magnetic field and the gyromagnetic ratio of the nucleus. The Larmor frequency is a key parameter in NMR experiments, as it determines the resonance condition for different nuclei. Understanding the Larmor frequency is crucial for designing and interpreting NMR experiments. Knowing the Larmor frequency allows us to properly excite and detect the nuclear spins in a sample. This slide introduces the concept of the resonance condition in nuclear magnetic resonance (NMR) spectroscopy. When the frequency of the applied radio waves matches the Larmor frequency of the nuclei absorb energy and transition to a higher energy state. This energy absorption causes the nuclear spins to align with the applied magnetic field, resulting in a change in their spin state. The transition of nuclei from the lower to higher energy state generates a detectable signal, which is the basis for NMR spectroscopy. The slide introduces the key relaxation processes in nuclear magnetic resonance (NMR) spectroscopy. Spin lattice relaxation involves the transfer of energy from excited nuclear spins to the surrounding lattice, allowing the system to return to equilibrium. Spin-spin relaxation refers to the transfer of energy between spins, leading to a decrease in the coherence of the spin system. The relaxation times, T1 and T2, characterize the rates of these relaxation processes. These fundamental relaxation processes are crucial for understanding the behavior of spin systems in NMR experiments. Chemical shift is a fundamental NMR parameter that reflects the electronic environment surrounding an atomic nucleus. Electron density around the nucleus affects the chemical shift. Higher electron density leads to shielding and lower resonance frequency, while lower electron density leads to deshielding and higher resonance frequency. Each type of nucleus in a molecule has a characteristic chemical shift, allowing identification of different functional groups and structural features. Analyzing chemical shift values provides insights into molecular structure, helps identify different types of protons or carbon atoms, and determines the presence of specific functional groups. Spin-spin coupling is an important phenomenon in NMR spectroscopy. It occurs when the magnetic field of one nucleus affects the spin state of a neighboring nucleus. The magnitude of this interaction is measured by the coupling constant, J. This provides valuable information about the relative positions and chemical environments of the coupled nuclei. Spin-spin coupling leads to signal splitting in NMR spectra. Instead of a single peak, we see a multiplet with a pattern of peaks. The number of peaks and the splitting pattern depend on the number of neighboring nuclei and their coupling constants. Understanding spin-spin coupling is crucial for interpreting complex NMR spectra and elucidating molecular structures. NMR spectroscopy relies on specialized equipment to generate and detect radio frequency signals. Key components include a powerful superconducting magnet, RF transmitter and receiver, sample probe, and computer system. Higher magnetic field strengths provide greater sensitivity and resolution in NMR measurements. The sample probe positions the sample in the magnetic field and enables interaction with RF pulses. The computer system controls the entire process, acquires and analyzes the data, and displays the resulting NMR spectra. Sample preparation is a crucial step in NMR spectroscopy as it ensures optimal signal acquisition and data interpretation. The choice of solvent, concentration, and temperature can significantly impact the quality and interpretability of the NMR spectra. Choosing a suitable solvent is crucial for solubility and spectral clarity. Appropriate sample concentration ensures adequate signal intensity. Temperature optimization enhances spectral resolution and peak sharpness. Removing dissolved oxygen through sample degassing minimizes signal broadening. Proper sample preparation is essential for obtaining reliable and interpretable NMR data. The 1D NMR experiment involves a series of radio frequency pulses applied to the sample. These pulses excite the nuclei, causing them to transition to higher energy levels. The excited nuclei then relax back to their ground state, emitting signals. These emitted signals are detected by a receiver coil and processed. The processed signals are finally displayed as a 1D NMR spectrum. 2D NMR is a powerful technique that provides detailed structural information about molecules. It correlates different NMR signals, allowing us to identify interactions and relationships within the molecule. There are various types of 2D NMR experiments, including COSY, NOESY, and HSQC. 2D NMR is widely used for structural determination of proteins, nucleic acids, small molecules, and complex mixtures. It plays a crucial role in chemistry, biology, and medicine. 2D NMR techniques provide a richer dataset compared to 1D NMR, enabling more accurate structural analysis and identification of complex molecular interactions. Homonuclear correlation spectroscopy, or COSY, is a powerful NMR technique that reveals connections between protons within a molecule. It does this by detecting spin-spin coupling interactions between coupled protons. This allows us to visualize the relationships and connectivity between different parts of the molecule. The COSY spectrum has diagonal peaks representing individual protons and off-diagonal cross peaks indicating which protons are coupled and connected. Understanding these molecular connections is crucial for elucidating the structure and behavior of complex organic compounds. The COSY experiment provides invaluable insights that complement other NMR techniques like 1D proton NMR. Heteronuclear correlation spectroscopy is a powerful set of NMR techniques that allow us to determine the connectivity between different atomic nuclei, such as protons and carbons or protons and nitrogens. The 1H13CHSQC experiment is a common method for identifying the direct connections between proton and carbon signals, which is crucial for structural elucidation and assignment of NMR spectra. The 1H15NHSQC experiment is used to correlate proton signals with nitrogen signals, particularly valuable in studying proteins and peptides, as it allows us to identify amino acid residues and their interactions. The HMBC experiment provides long-range correlations between protons and carbons, revealing connectivity patterns that may not be readily observable in other experiments. These techniques are essential for analyzing complex molecules, such as biomolecules and polymers, as they provide valuable information about structure, dynamics, and interactions. Solid-state NMR is a powerful analytical technique that allows us to study the structure and dynamics of materials in their solid state. It provides unique insights into the interactions between molecules, which is crucial for understanding material science, chemistry, and biology. Key differences from solution-state NMR. Solid-state NMR is used to analyze materials in their solid form, while solution-state NMR is used for materials in solution. Solid-state NMR spectra are broader and more complex due to the restricted motion of molecules in solids. Solid-state NMR techniques are used to investigate a wide range of solid materials like polymers, proteins, and ceramics. Solid-state NMR gives us a window into the properties of solid materials that can't be easily studied using other techniques. Nuclear magnetic resonance, or NMR, is a powerful analytical technique with a wide range of applications. One key application is structural elucidation, using NMR to determine the structure of molecules, including complex biomolecules like proteins and nucleic acids. NMR is also widely used in metabolomics, analyzing the concentrations of metabolites in organisms and cells to study their metabolism. In material science, NMR can provide insights into the structure and dynamics of materials like polymers, ceramics, and composites. Perhaps the most well-known medical application of NMR principles is magnetic resonance imaging, or MRI, which is a crucial diagnostic imaging technique. NMR spectroscopy is a powerful analytical technique that provides detailed insights into the structure and dynamics of various molecules, from proteins to small molecules and polymers. When studying protein structure, NMR can reveal the three-dimensional folded state of the protein, as well as information about its dynamics and flexibility. For small molecules like drugs, NMR can be used to determine the conformation, or 3D shape, of the molecule by analyzing the coupling between different nuclei and the chemical shifts observed. NMR is also valuable for characterizing the architecture of polymers, including their chain lengths, branching patterns, and tacticity, the relative orientation of substituents. Additionally, NMR is a key tool for studying intermolecular interactions, such as protein-ligand binding. It can provide insights into binding affinities and the specific modes of interaction between the molecules. Overall, NMR is an indispensable technique in structural elucidation across a wide range of molecules and systems. NMR is a powerful tool for identifying and quantifying metabolites in biological samples like blood, urine, or tissue. Analyzing metabolic profiles can help diagnose diseases like cancer, diabetes, and cardiovascular disease by detecting changes in metabolite levels. NMR can also be used to study the effects of drugs on metabolism and identify potential drug targets. Additionally, NMR provides insights into the regulation of metabolic pathways and how they are affected by disease or environmental factors. This slide summarizes the key points about the future of NMR spectroscopy. NMR has become an indispensable tool in many scientific fields, from structural analysis to metabolomics and materials science. Looking ahead, the focus will be on improving the sensitivity, resolution, and automation of NMR experiments. Advances in NMR methods will enable deeper insights into complex biological systems and materials. The continued development of NMR technology will expand its applications and impact across a wide range of disciplines. In conclusion, NMR spectroscopy has a bright future as a powerful analytical technique that will drive scientific discovery in the years to come.